So if you feel uncomfortable with that, so feel free to turn off your camera. All right. Oh. So this symposium series is being organized by the Plant.ID network, which is an MSc and Marie Curie Actions, ITN, funded by the European Union. And its core is comprised of 15 PhD students, which are listed here below. That is me. I'm your host for today. And 2 other PhD fellows of the network will be presenting their research today. The first 2 talks, actually. And so all the Plant.ID fellows are dealing with subjects related to molecular identification of plants. Short word about myself. Who is that guy behind the screen? Huh? I'm doing my PhD at the School of Biology of the Aristotle University of Thessaloniki, in association with the Institute for Applied Biosciences at the Center for Research and Technology in Thessaloniki, Greece. And I'm dealing with the traceability of species of the Greek flora in food and medicine through DNA technologies, which are DNA metabarcoding for authentication, barcoding high resolution melting, and targeted sequence capture for species limitation in Greek salad orchids. My supervisors are Andreas Druzas and Panagiotis Madesis. So we published recently a paper. So if you want to check it out, so you can find it here in the corner below. It's pretty interesting. But enough advertising. Let's go back to that to the series. So molecular monitoring. That's the subject we're going to discuss today. That's the program. So first we'll have a presentation by Ozan, and followed by one of Felicitas, our 2 PhD fellows of today. That will be followed by a short break, a 15 minutes break. And then please stay tuned for our 2 keynote speakers, Joanna Freeland and Anthony Booker, which I'm personally very looking forward to. And that will be in the second part of this symposium. Some general remarks I would like to share. Please mute your microphone during the talks for obvious reasons, and keep your videos on as well, because this, we believe, it creates a more pleasant atmosphere. Every talk will be followed by a 5-minute question round. So if you would like to ask questions or raise discussion points, please do so by using the chat function below, or raise your hand. So with this, I would like first speaker. Our first speaker is Ozan, one of our PhD fellows within the Plant.ID network. He's based in BaseClear in Leiden, Netherlands. Ozan is a former marine biologist working at the Institute of Marine Sciences of the Middle East Technical University, Turkey, where he also obtained his master's degree. He gained experience in molecular techniques such as DNA barcoding, eDNA metabarcoding, and phylogenetic approaches for marine biodiversity assessments, and worked after his master's at the Evolutionary Genetics Laboratory at Ankara University. Within his PhD project, he's exploring genomic approaches for freshwater quality assessments by identification and quantification of bioindicator diatom complexes. So such new methodologies are necessary given the current anthropogenic threats to aquatic ecosystems and the challenges of current methods. So today, Ozan will tell us more how he tackled those issues with his project. I will be discussing his preliminary project results. I would say, Ozan, the floor is yours.
Thank you, Bastien. Can I
will,
now I will share my screen. Can you see it?
Yes. All right.
Okay.
Good luck.
Okay, thanks, Bastien, and I want to thank you all also for joining our seminar series. And now I will try to explain my research study, what I did in the last 3 years. To start with, I am Ozan, and I am doing my PhD in a DNA sequencing company called BaseClear. And this is a collaboration between Naturalist Biodiversity Center, BaseClear, and Leiden University. And as you can see, I changed the title of my presentation because we are performing 2 studies, both identification and quantification, but the quantification part is still going on, and I couldn't get the results that I was expecting to get until this presentation. So I will present the first part, identification of Nitschia palea. So title of my presentation is Species Delimitation in Taxonomically Problematic Bioindicator Diatom Nitschia palea. My main supervisors are Barbara Gravendiel and Peter van Bodegom from Naturalist Biodiversity Center and Leiden University. So to start with, I want to give some brief information for the people who are not familiar with diatoms in general. So as a general information, diatoms are photosynthetic macroalgae. They are living in aquatic habitats, and they have a significant contribution to primary production. They are photosynthetic, and they also have silica cell walls. There is a huge diversity of diatoms. And there are around 200,000 living species that are described currently, and there are 100,000 more in the fossil record. And different species of diatoms have taxon-specific ecological preferences and tolerances. So that means when the water is polluted, sensitive taxa decreases in abundance and tolerant taxa increases in abundance. And this is causing a change in the community composition of diatoms. And this is our water quality assessment. This is a method to perform water quality assessment also. And in the species checklist, diatom species checklist of the Netherlands, each diatom species is described with different ecological indicator values. And here, as you can see, Nitschia palea is described from higher saprobit and trophic state levels, which means that they are more abundant in comparatively polluted waters. And there is another group, Variety debilis. It is described from comparatively cleaner oligotrophic waters with lower values. And the main taxonomic problem in this species complex is this. And these different varieties have slight differences in their morphology, mainly in their width and stria density. And also, based on our communication with diatom experts, the narrower outline shapes are more common in comparatively cleaner waters, while in polluted waters, and by pollution here, I mostly mean organic pollution and heavy metal pollution. And in polluted waters, their outlines are comparatively broader. And there are also previous studies, where they try to delimit those taxa using morphological and molecular data. And with morphology, they have a significant overlap, so it is not easy to separate them. And with DNA, there is insufficient resolution with conventional markers. So currently, both of those varieties and these groups are treated as a single group in water quality assessment. So they are assigned a single ecological indicator value, which is kind of preventing the recognition of different ecotypes. So what we did was we ordered 10 strains, 10 Nitschia palea strains in total, 4 from Belgium coordinated collection of microorganisms, and 6 strains from Tonon culture collection in France. I grow them in the growth chambers of Naturalist Biodiversity Center. And here it is not really clear, but on the left picture, you can see the colonies that they are forming biofilms at the bottom of the Petri dish. And also in the flask, they are attaching to the sites and they are forming like a brownish biofilm. And in this process, growing them to enough amount takes several months to reach enough amount. And after that, simply by centrifuging, I am collecting all the cells in smaller tubes to perform further studies on them. And during their growth, we need to screen their health also. So I transferred these cultures every 1 to 2 weeks to new medium, and I collected small samples to screen their health. And when there are different methods to observe diatom cells, when observing living cells, we need something like a differential interference because we need a black, like a darker background with the contrast. Here you can see that they have two plastids, and the parts that are shining are their plastids actually here in the left part. But here, when we observe them like this, their valve characteristics are not visible, and their taxonomy is usually done with those valve characteristics. So I also prepared oxidized valves to screen them. And also in the beginning of my project, our aim was to collect environmental strains, like isolate Nitschia strains from environmental samples, grow them, and study on them. But mostly due to Corona, we couldn't perform those studies. But just to demonstrate, here two Nitschia studies that we found from biofilm samples that we collected from Netherlands. And they are present with different types of species in different waters. And then we also collected scanning electron microscopy images to go a bit deeper. Here, as you can see, most of the features are visible, and we can measure and count the main characters to see if these individuals, these cultures, strains, really belong to the correct species. And on the left part, on the up, you can see the outer side of the valve, and in the right one, you can see the inner valve. So normally these are attached like a petri dish together, and one of the valves is smaller than the other. And with each cell division, they are becoming smaller. And when they reach their size limit, smaller size limits, sexual reproduction is triggered. So they are producing the longer initial cell again. And here in the bottom picture, you can see an unseparated valve. Those are two valves attached to each other. But this is a comparatively small, shorter individual. And mostly the taxonomic identification is done by measuring and counting these characters. These images were collected in Naturalis Biodiversity Center again by Bertie John using a scanning electron microscopy system. And using these images, first I performed the shape outline analysis. To do this, first we processed raw images collected from scanning electron microscopy, and we converted them to, at first we extracted individual cells from each image. We collected 50 images per strain. And after extracting that from each image, I filled them with black color and exported them on white background so we can study the shape outline. And there is a package, morphometrics analysis package in RStudio. I use that package. Here you can see the piled overview of all images. There are around 50 images per strain. And here, one thing that I want to mention is that for the last strain, 907, this strain was unhealthy when it arrived. Most of the individuals were already, their shapes were deformed. So I couldn't extract and use the outlines for this strain. There are only 4 images for that. And there is one more thing, this TCC 13903. There are 2 different visit ranges, as you can see here. And that is mainly because this strain is capable of self-reproduction. This is an information that we got from the source collection. And as I mentioned, their length changed during life cycle, but their width remains more or less constant. So when there are 2 different width ranges, that is like an indication of 2 different clones. But here, that strain is capable. Normally, Nitschia is described as, it can't self-reproduce most of the strains, but that specific strain was self-reproducible. So at some point during its life cycle, it reproduced and produced the longer cells. So this is why we had 2 width ranges in that specific culture. And after collecting these outlines, I used an analysis transformation called elliptical Fourier transformation. This is simply like a curve fitting model a mathematical approximation, and it converts XY coordinates into coefficients, 4 coefficients in total, and describes the outline based on that 4 coefficients. And the number of harmonics describes how many times that coefficients needs to be calculated. Like for 5 harmonics, for example, there are 20 coefficients in total, and so on. So using that different number of harmonics, and there is one more thing I should mention here, that the valence in diatoms is a taxonomically important feature. Based on that shape of the valence, different species are described. So that's an important character, and I wanted to check if we can capture the outline shape at the valence correctly using this transformation. So I mapped different approximations, different models on the original outline. And as you can see, until here, until 17, for example, the neck, there is a neck in the valence, but until 17, that neck is not represented. Like here in blue, it is going straight, and the inflation at the end is not really represented. So I increased the number, and I used 40 for this analysis, 40 harmonics for transformation. And then using that coefficients, I performed the PCA analysis to look at the variation in their shape outline in general. Here, the colors are a bit within each other. It is not easy to evaluate, but first I want to explain the axis. Here, the first PC1 axis is describing the width, like here from right hand, narrow outlines, it is going to the broader outlines on the left. And the second axis is describing how much the ends are protracted. Like here, you can see the ends at the bottom, ends are more clearly represented, but at the top, it is just straight, like an ellipse. So most of the variation is explained by the first axis, 98% 2. And also there is no clustering based on second axis. But based on this figure, we can broadly categorize them into 2 clusters where there is really small overlap between them. The first one on the right can be categorized like from linear to narrow to linear outlines almost, and the left part, second cluster, is from linear to broad outlines. So after this, at this point in the analysis, I just categorized them based on their width, because this was our first hypothesis, like the species in comparatively cleaner waters have narrower outlines. So I separated them based on that broadly. And I made a color gradient from narrow to broader outlines, and I used that color code for the next step. Here you can see the morphometric data and morphometric results now. As I mentioned, these are ordered from narrower outlines to broader outlines from light green to dark green. And I excluded one strain, 907, because there were only 4 outlines for that strain. And as you can see, length-width ratio is highly correlated with that color gradient. It is going down as it gets darker, mainly because shape outline is most correlated with shape length-width ratios. The other characters that we measured here are mostly for taxonomy, like their width, for example, all fall in the range, correct range that is given for this specific species. And looking at their stria densities, there are 2 strains which were deformed and not healthy. So this is also an issue that can be seen when we look at the cultures. Their outlines were deformed. And the other characters are also fall in the expected range. Now I am going to use this data to make some further analysis, like I will check if the characters are correlated with each other or not. And also we are going to discuss this with diatom taxonomists to look for more detailed characters that we can identify to separate those 2 clades. And this is the morphological part that we are performing. And as a second part, we are also performing molecular analysis. I also extracted RNA from all those strains, all those cultures. And actually in the first picture, you saw that there was a larger flask and also Petri dish. I am using the samples growing in Petri dishes for microscopy analysis, because for RNA sequencing, we need much more cells. Actually, mostly in the kits, they are suggesting like 5 to 10 million cells in total. So in Petri dish, they were not growing in enough amounts, so I grow them in bigger flasks. And I also counted their screen their growth to see how much size we can obtain in the end. And I did that using a Bayer counting chamber that I counted the cells every day for a month. And for this molecular analysis, we only use the 4 strains coming from BCCM, Belgium Culture Collection. The TCC samples are currently being analyzed, so everything I show about molecular part will be on those 4 strains only. And based on this growth curves, we found out that we can grow 3 to 6 million cells in 100 ml cultures. And then I performed RNA isolations using Kaijin plant minikit with some modifications because they have silica cell wall and it is a bit harder to externalize the cells compared to plant cells. So there were some modifications like liquid nitrogen and so. And with those modifications, we isolated RNAs and we sequenced them on a NovaSeq platform in BaseClear, pair-length sequencing. And the results for 4 strains are ready, and the remaining TCC samples are being processed right now. And this is my transcriptome assembly pipeline. For this part, I am working together with Andrew Walverson from University of Arkansas. We are constructing the pipeline as a collaboration. And mainly the first orange part is pre-processing reads. There is some quality check, error correction, and trimming. And at the end, right now I am trying to find nuclear phylogeny. So I am filtering out ribosomal RNA, plasmid, and mitochondrial sequences also. And after that step, there is assembly. And finally, there is post-processing after transcriptome assembly, like assessing the quality, predicting proteins. And finally, I will try to find orthologs in those transcriptomes and build a phylogeny based on ortholog sequences. So I am using a software called OrthoFinder, which gives gene trees but based on amino acid sequences, not coding sequences. And here I am working on a species which is recently diverging, like all the strains are from a single species, and we know that some strains are in different ecological conditions. So it's a recently diverging species. So I will use DNA data instead of amino acid sequences. And in the pipeline, we built homolog coding sequences, gene trees in the end. And from that point, I will use another phylogenomic pipeline to infer orthologs to separate paralogs from orthologs. And then only using the orthologs, I will try to estimate species tree using both concatenation and coalescent-based methods, Astral and Raxaman. And in the end, we will compare that data with our morphological results to see if we can provide further evidence on species delimitation in this complex. And as a small side study, I also extracted the sequences that was mapping onto plasmid and mitochondrial genomes and Silva database, ribosomal RNA database, and I assembled those sequences from those reads to see if we can correctly identify the same samples using molecular data. And here again, we analyzed only 4 samples. So these are the results coming from 4 samples, 91, 92, 94, and 751 are my samples. And as you can see, we found like a perfect match with the other strains. These are actually the same strains in the collection. This data was collected in previous studies. So we can also prove that our strains are coming from the correct source. And this is it for that part. So to make a conclusion and to summarize, my research questions in general are if there are morphological characters or morphometric methods like shape outline analysis that can be used to further investigate the species taxon delimitation in this complex. I know another part is if we can use orthologs also to improve taxon delimitation in the complex, because with standard markers, as I mentioned, there is not enough resolution and so on. But my main research question in the end is if those taxa do really have different ecological preferences because this is the part where we can really improve water quality assessment or provide some evidence or data for that part. So with the first results, we have some preliminary evidence for 2 clades, but both morphological and molecular data is showing that. But in the previous studies with larger data sets, there is evidence for like 3 different lineages in the complex. And right now I am assembling the transcriptomes, and with the further statistical analysis on morphometric data, I will try to conclude and put them all in an article to publish, and probably around the end of this summer. And we also have a quantification study, as I mentioned, that's also going on. We are preparing mock communities from different strains. I used TCC samples and prepared mock communities in different amounts. And we are using a method called multi-species genotyping by sequencing to quantify the abundance of those different strains in mock communities. And this is all for my PhD project. Thank you for listening. I hope you enjoyed it. And if you want to contact me further, or if you have any other questions, you can always contact me from this email address. And I want to thank you all. Bastien, is he frozen? Yeah.
I think that Bastien's having just a couple of computer problems with his right now. But that was a very nice talk. Thank you so much. Are there any questions? No? Then I think we can move on to Feli, perhaps.
Yes.
And so Feli is a current student in the Plant.ID network as well. She is based here at the University of Oslo. She works in the pharmaceutical department, and we are really looking forward to her talk.
All right. Thank you, Marcella. I can just introduce myself shortly, maybe about my background. So I have a background in molecular ecology and as a horticulturalist, and I specifically focus on medicinal and aromatic plants. And yeah, today I want to introduce you to the Herb Farm project, which is an integrated, it's on play, right? Yeah. An integrated study to authenticate Chinese herbal drugs. And the main objective is the development of an authentication protocol for Chinese herbal drug formulae available on the European market. And I focus on common herbal preparations for chronic female disorders. It's an integrative approach, so I focus on the purity, which means species identification and quantification. I use methods like DNA barcoding, multi-loci metabarcoding, and chemical fingerprinting. And I also conduct efficacy and quality testing with the detection of bioactive metabolites and bioactivity assays. Yeah. Today's course is specifically on the authentication of TCMs for women's healthcare on the European market. Let's go. In contemporary gynecology, we observe that there's an increased application of herbal medicine therapies in women's healthcare. And this is most likely due to the fact that there are fewer adverse reactions than, for example, in hormone replacement therapies, and it displays a non-surgical option. And we look at 2 TCM formulae, which are frequently prescribed or sold as patent medicines for treating gynecological conditions related to major dysharmonias and chronic pathogenesis, for example, dysmenorrhea, abdominal pain, or infertility. This is what I call the Wild East-West global herb trade. So I just want to give you an impression about my sample collection. I work with different samples, which are pills, green pills, or tablets and granules, and also crude herbal material. And for some of these samples, I actually don't have a precise ingredient list. And these products are freely available on online shops. And as you can see here in the Petri dish, it is supposed to be a single species herbal batch, but that's questionable, as we can see with the mere eye that there are different pieces of different species. And I also was lucky to have a look into the Chinese herbal medicine collection of Kew Gardens. And it was really interesting to see what they have received from custom, like different colored herbal products, patent medicines over the years, and also different CITES listed species. This supports relevancy for herb form. And it's interesting that the average European TCM practitioner uses over 300 herbal medicine ingredients in their daily practice. Yeah, and as Nature also stated, and there are different articles out there, that many of those TCMs contain species which are not listed in the ingredients. And most of you have heard of aristolochic incidences, which pose a serious health risk to the public. So yeah, so I want to just quickly really give you a background about the formulating, since this is a foundation for this project. And for the sample collection, Chinese herbal medicine is based on multi-herbal formulas, and synergistic effects are also attributed to those formulas. As such, I analyze and elaborate Gucci Fuling 1, which is also called the cinnamon twig and Poria cocospur. And it's one of the classic Chinese herbal formula for female disease patterns. And it was already developed 1,800 years ago. And it is still listed as it was developed back then in the Chinese Pharmacopoeia with 5 species. And it's one of the most frequently prescribed Chinese herbal formula in the management of uterine fibroids. Just some undermining kind of reasons to choose this formula. It has been shown to be quite effective. And there's one study which is very interesting. It looks at different in vivo experiments for the analysis of its effects on uterine fibroids and also sex hormones. And on the right-hand side, you see a network of the herb pairs which are most frequently prescribed for formula in gynecology. And based on that herb pairing and some background, I have formulated the second formula, which is more specifically created for a specific disease pattern, which is called endometriosis. So this is quickly the first formula, which comprises cinnamon, Poria, peony, 2 different species, and Persica semen. And also the second formula, which interestingly incorporates feces from the flying squirrels, and also the Angelica sinensis Chinese Angelica root, which is also in European medicine known for gynecology, gynecological treatments. Now we come to the first part of my results, and I want to introduce you and show you the results for my applied DNA barcoding study. So the hypothesis here is that Sanger sequencing techniques can create problems for the authentication of highly processed herbal materials. And as you see here from the example, we look at different and 2 comparative electropherograms, which are mixed. So I have not clear peaks signaling the nucleotides. And generally, I just want to say that I conducted a quite harsh quality control measure for the identification of my sequence data. And yeah, I look at the putative ingredient. This is my goal, like which ingredients can really be identified with certainty. And from this analysis, we could actually conclude that RBCL is not adequate for putative ingredient identification for our samples, since we don't have any identification on species level. Quickly, on the right-hand side, like, I identified on species level with a 98% threshold, and a variation within the species or deviation of 1%. And before that, I want to mention that the identification is based on the base calling quality and the identity threshold. As a result, we can actually kind of conclude, as I state here, that ITS and ITS2 can possibly be used for a combined discriminatory power for identifying specific species, for example, Peonia fruticosa. This might be interesting for further studies and an improvement of applied barcoding using Sanger sequencing techniques. But yes, it's kind of clear, or it is clear, that ITS2 is the most effective barcode here, which is also the standard barcode for the authentication of medicinal plants in national pharmacopoeias. This was kind of assuring, but now I want to just, from my personal experience working with natural products, here we look at a pile of cinnamon, and it was interesting that actually in none of my results I could detect any cinnamon. Oops. Nah. And I had cinnamon in my nose for a whole week, and I got quite suspicious about my results. And that's why I want to quote that humans have been selecting medicinal plants and foods based on organoleptic assessments of suitability and quality for thousands of years. And that's what I kept in mind thinking about the considerations for this applied DNA barcoding approach. So I work with highly degraded DNA from processed samples, obviously. In Chinese medicine, there's a processing called Pouchy, which is maybe an important information here. And I also have inhibitory contaminants in the samples, like polysaccharide salts and phenols. And of course, there's a possibility for multi-species samples, or at least a contamination. And that's what we clearly saw also in one of the examples that I work with natural products. And there's a natural variation included in that. As a conclusion, the conventional and standard markers are not discriminative enough for my species or sample collection. As a result, I verify this study with HPTSC, chromatographic verification, and this is crucial. For each species, we have a different methodology. So this means I have 14 different methods. And also, we work with a multi-loci metabarcoding for complex herbal materials, which will give us more information on the ingredients. This is for my chromatogram. To give you an example, this one is Peonia lactiflora, and here you can see that peoniflorine is the chemical reference compound, and also we work with a botanical reference compound. And in all the samples except the fourth one, we actually can detect peoniflorine, which speaks highly for the identification of Peonia lactiflora. I want to show you another example for these results. Now my laptop is stuck. Is this see it? No. We're back, right?
We're back. Good.
Okay. Computer got stuck. This is another example for Peony subverticosa. And here, first of all, I want to point out that there is much confusion about the nomenclature of Peony subverticosa. So that's an open question still, how I will include that in my analysis. But anyway, peonofluorine and peonol are combined chemical reference standards here. And we can see that in all the samples, we actually detect both bands of both chemical reference compounds. And the mixtures, the first and second formula, don't reveal peonol. So we assume or suspect that Peony subverticosa has been substituted with additional Peony lactifluorine. That's a high possibility. And now I quickly want to also show you the results from my quality testing. So I conducted immune activating, or I'm conducting a bioequivalence study comparing traditional decoctions with granules and assess the immune activating and anti-inflammatory effects. And here I only focus on one formula, Gucci-Fuling-1, which is the first classical formula. And I work with bioassays, cell enzyme-based in vitro screening and radical scavenging, and also with HPLC for quantification of the bioactive compounds. And these are the results for the NO inhibition assay in a dendritic cell line. So here I can test anti-inflammatory effects and immune responses of my extracts. And what is interesting that mostly for cinnamon and peony, I have a strong NO inhibition in higher concentrations. And also the mixtures are stronger NO inhibitors. And that's kind of nice because it reveals that the combination of different combination of different plant species might have synergistic effects, and that's kind of the recipe for Chinese medicine. This is the xanthine oxidase inhibition assay, or the results for it. And here I measure the inhibition of the extracts on pro-oxidative enzyme, xanthine oxidase. And also here we see that cinnamon and both peony species have a high inhibition effect on this particular enzyme. And then I have a DPPH radical scavenging. And here I support the results and the findings from the last assays that actually cinnamon and peony have a strong radical scavenging effect, and also the mixtures are actually the strongest radical scavengers. Yeah. So thank you very much for your attention, and I want to thank especially my group, the team around Perform, Helle, Hugo, Christine, and Caroline, and special thanks to Francesca, Scotty at UCL, who works with me on all the chromatography. So big thanks to you, Francesca. Thank you.
All right, thanks, Feli, for this nice, good-looking presentation, for your interesting talk. I'm sorry for the technical issues we had, but I think now we will have some time for questions. Someone has a question or a discussion point that you want to raise?
Hi, Feli.
Hi, Tony.
Hi there. Thanks for the presentation. It's very nice. I was just wondering, looking further down the line, how do you think this work is going to affect Chinese medicine? Where do you see its its main application, given that most companies do their own in-house quality testing and authentication. Where do you see the need for this kind of work?
Thanks for your question. That's a good question. So I thought about that myself quite a lot, and really my main purpose with this work is also to create awareness amongst practitioners and patients, because there's a risk for adulteration and substitution, and also unknown fillers which might cause health risks. And that's yeah, I think creating awareness in terms of really making people aware of where they source their material, because sourcing is part of the whole process of gathering and using herbal medicine as a treatment option, and yeah, having high quality and plant material. And the methods which I'm using, for example, or the ones which I also introduced you to now, for example, applied DNA barcoding with means of Sanger sequencing and ITS2, is something regulative authorities do already partly in different countries. And yeah, I think it is really important that we have a close look at the methodology. Is it sufficient enough to assure the quality of these products? So that's why I think, yeah, what I want to point out with that.
Okay, thanks.
Thank you.
There is one more question. I think we have time for one more question. There is a question in the chat from Kenny. Did you look at any other markers than the reported RBCL and ITS? Now, how about MET-K, PSBA, TRNH, and or concatenating marker regions?
So we looked at MET-K also, and this was preliminary excluded from the species analysis, really due to very low species resolution and bad amplification. And also, yeah, I think that's an answer to this question. And I think what I want to say here, because what I showed now are really applied methods. However, for example, with our multi-loci metabarcoding study, which is, by the way, all sequenced and in the process of being analyzed, that's something which is an asset to the usual authentication methodology, which is used for regulative purposes and also control purposes. And that should be considered, I think, in future, that there are options to scale up for authentication and quality control purposes, amongst other measures, of course. Yeah.
All right. So I think now we'll have a short break. I propose I will be back at 15:05. That will be approximately 13 minutes break. So I would say grab a cookie, grab a coffee, and we'll be back in a couple of minutes with keynote speakers, Joanna Freeland and Anthony Booker. So stay tuned. Hello, welcome, Joanna.
Hello, I see you.
Nice to see you, yeah, in person.
Yes, semi in person.
Exactly, semi in person, yes.
Do you want me to share my screen or?
We'll start in 3 minutes because it got delayed a little bit. So in 1 minute, actually. So we'll just wait until people are coming back and then I'll give a quick introduction. So what, yeah. Just give it 1 more minute and then we can start. All right, welcome back, everyone. We'll proceed with our next speaker of today. It's Joanna Freeland, full professor at the Department of Biology of Trent University, Canada, leading the Molecular Ecology Lab. Joanna has published widely in the area of molecular ecology and in recent years has been particularly interested in hybridization and invasive macrophytes. Joanna is author of the book Molecular Ecology, which discusses the increasing prevalent use of molecular data and ecological studies, providing an up-to-date account of the field as it is today. She's currently also working as an associated editor of Plant Ecology and Diversity. Her past and current research programs use molecular genetic data to address questions such as the relationship between environmental change and genetic diversity, the taxonomic status of endemic fauna, the extent to which populations are interconnected by gene flow, and the importance of genetic lineage to the invasibility of plant populations. In her presentation today, Joanna will talk in more detail about which molecular tools are currently being used for early detection of invasive aquatic plants and identification of cryptic invasive macrophytes. With that, Joanna, the floor is yours. I'll stop sharing my screen so you can share yours.
All right, I'm just going to share my screen here. Okay, well, thanks very much, and thanks for inviting me to participate in this seminar. I won't repeat the title because it was you just heard it. But I do want to acknowledge that what I'm talking about today is, of course, not just my own work. It's work that I've done with a lot of collaborators, some of whom I've listed here. So keeping with the theme of this seminar series, Plant.ID, I wanted to focus on identifying invasive species. And of course, I don't need to tell this audience the problems that we have with invasive species. We know they're increasingly widespread, decimating ecosystems around the world, and they span a wide range of taxonomic groups, virtually every taxonomic group. Today, I'm going to focus on freshwater ecosystems, which are biodiversity hotspots around the world, but they're also among the most threatened ecosystems in the world. And a major threat to them is biological invasions, which again spans multiple taxonomic groups, but I'm focusing on invasions by plants, which can be hugely problematic in these habitats and ecosystems. They reduce native biodiversity, they can alter nutrient cycling, reduce light penetration through the water column, alter water cycling and water chemistry, impede recreational access, and so on and so forth. So basically, they're spreading, they're getting into these sites, and they're really altering the sites and doing a lot of damage. Now, I think often when people think about invasive plants in a habitat, they think, okay, it's pretty obvious. A new species has come in, it's starting to dominate this habitat. As you see here, this is a photo of floating yellowheart, which is an invasive species in North America. But the reality is it's not always that obvious. So I wanted to begin by talking about some of the challenges surrounding cryptic invasive taxa. And cryptic invaders can include species. So this would be introduced species that morphologically resemble native species. They can include intraspecific lineages, so lineages of a species that is native, but those lineages arose elsewhere. And also novel hybrids, which are often hybrids between native and introduced species. It's fair to say, it's not too much of a sweeping statement to say that cryptic invasions in plants have been very poorly studied. And in fact, a recent published paper concluded that across taxa, less than 1% of studies on biological invasions included cryptic species, for an obvious reason is being that we often don't know that they're there. When I think about using molecular markers to identify these cryptic invaders, there's really two steps. The first is to identify them, and then the second is, once you know that they are there, to try and find out where they are so that you can make connections between these invaders and dispersal, range expansion, effects on habitats, and so on. But note that you often need different sets of markers for these two steps. Now, I'm going to talk about a couple of case studies. And these, I know that most of the audience is in Europe. These are both examples of plants that are native to Europe, but are invasive in North America. And the first one is Phragmites australis. And about 20 years ago, this paper up here is published by Saltonstall, identified that we actually have an invasive lineage of Phragmites in North America, even though we also have a native lineage. So the native one is on the left here. This is a photo from southern New Brunswick. And on the right, this is the invasive one. This is a photo from southern Ontario. And this was initially identified using a phylogeographic study using chloroplast sequences, and has since been supported by other lines of evidence, microsatellite data, morphological data, ecological data, and so on. And so we now conclude that there's actually two subspecies of Phragmites australis in North America. The subspecies australis, which is the one that was introduced from Eurasia, probably from Europe, and that's the invasive one. And then we also have the native subspecies Americanus. So we already know that there is this cryptic subspecies here. That's been identified. A lot of research has been done on this. But recall that cryptic invaders can also include novel hybrids. And there is a concern now that there may be hybridization going on between these two subspecies. This was documented a few years ago in a restricted geographical area near Las Vegas, another publication from Saltonstall et al. My neck of the woods is around the Great Lakes, the Laurentian Great Lakes region. And in the past couple of years, a lot of conservation managers have approached me and said, we're looking at these stands of Phragmites. They appear to be the native phenotype, but they seem to be growing in a manner that's more consistent with the invasive subspecies. So we're a little bit concerned that these might be hybrids. Now, there already exist chloroplast markers that can differentiate the two subspecies, but of course, they don't identify hybrids because as with most angiosperms, chloroplast DNA is maternally inherited. So if you just have data from the chloroplast DNA, all that's telling you is who the mom was. So that's not going to identify hybrids. We have in the past identified hybrids or likely hybrids from microsatellites because allele frequencies differ between the subspecies. There aren't clear-cut species-specific alleles, but the frequencies differ. So we can identify them this way. This is an expensive approach. It's time-consuming. It can be challenging, partly because these plants are polyploid, which makes it much harder to do things like identify null alleles. And of course, you need reference databases. You can't just get data from one plant and say that's a hybrid. You have to compare its genotype to allele frequencies of known native and known invasive subspecies in this area. So it can be done, and once you get those data, use something like structure analyses or principal components analysis and conclude that yes, these are probable hybrids. But as I say, very expensive, time consuming, tricky, repeatability between labs can be problematic. So the conclusion so far is the hybrids are rare in some places. They've been identified as invasive in this area around Las Vegas. But the question mark is, are they becoming increasingly common elsewhere? But so far, it's been very difficult to test. I'm just going to, and it's important to know this because a lot of conservation managers are on the ground actively trying to remove Phragmites from sites where they are taking over. And they want to make sure they're not removing the native subspecies. And they also want to know if anything's changing on the ground. Is this, state of play changing by the spread of potentially invasive hybrids? So this is very old school, actually. I know this seminar is more about new methods, but this is a very old school approach to get a very quick and simple screen to test for hybrids. And it's a PCR RFLP. And for the early career researchers, you may be less familiar with these. It's the simplest technique. Once you've compared sequences, and if you identify a SNP or an indel that is fixed in one of your target groups and not in the other, and if that mutation is within a restriction enzyme recognition site, then what you can do is just design primers, amplify that fragment of DNA, and then do a digest with a restriction enzyme. And if the site is present, it will digest, and if the site is absent, it won't digest. So that's what you see here. This is a paper. It's not been published yet. It's under review, so I won't go into details, but it's just showing that this is the introduced, there's the undigested and the digested. It does not have the restriction enzyme recognition site, so it doesn't digest. The native does have it, so it does digest. You see two bands, and the hybrid, because it has parental genes from each subspecies, you get both the digested and the undigested, and there you have it. So this is a really quick, straightforward, fast, relatively cheap method to quickly screen wide areas to find out if this invasive hybrid is around there. So we got very excited about this approach, and it's tying in well with a lot of on-the-ground managers. But I'm going to give you another example now using a similar approach that shows that it can actually be a little bit more complicated. And in this example, I'm talking about typha, which in North America we call cattails. They're often called bulrushes in England. There's lots of different common names. I'll just refer to them as typha. In Canada and the northern US, we have two species. There's the native broadleaf cattail, Typha latifolia, and then there's Typha angustifolia, which is the narrow leaf, which was introduced, although likely introduced several centuries ago, probably with early European colonizers. And in many places where they coexist, they rarely or never hybridize. But in the Great Lakes region here, they hybridize like crazy, and their hybrids are very invasive. They're hugely problematic. They reduce native biodiversity, they alter nutrient cycling in wetlands, they create huge amounts of leaf litter, and they really do change the landscape. So they are considered problematic. And again, with management and with understanding how these hybrids are altering the landscape, we need to be able to identify them. But it can be, even though the two parent species look really quite different from each other, it can be very difficult to identify the hybrids based just on morphological features, partly because they can they don't just create F1 hybrids. They can also create advanced generation hybrids, and they can also back cross to the parent species. Now, there's a lot of information on this slide. You don't need to know all the ins and outs. I don't have time to go through all this. And you can see from the bottom here, this understanding is based on a number of years and a number of investigations. But the sort of take-home message here is we know what the crosses are. We know things like the creation of F1 hybrids is asymmetric. So it's almost always Typholatifolia pollinating Angustifolia. Angustifolia is the maternal species to create F1 hybrids. And we know that there's also asymmetry when they back cross to Latifolia. It can go either way when they back cross to Angustifolia. And we also know that the F1 hybrids are fertile and they can produce advanced generation hybrids. So against this backdrop, obviously things become more complicated. We can, again, use microsatellites to identify hybrids. It's a bit easier than in Phragmites because they're diploid and because a few loci do have species-specific alleles, but it's still a relatively time-consuming and expensive method, particularly when you want to survey wide areas and lots of wetlands. There already exists, and it's been published in this reference here, a PCR RFLP test that can differentiate the chloroplast DNA of Angustifolia and Latifolia. But again, the chloroplast DNA is maternally inherited, so the chloroplast DNA will identify only the female parent. It won't tell us if it's a hybrid. So what we're currently working on is, can we come up with, and will it be useful to do a similar nuclear PCR RFLP test to be used in conjunction with the chloroplast PCR RFLP? And what we have to be aware of is, in this much more complex hybrid system, it's not going to identify all of the back-crossed and advanced generation hybrids. And I will illustrate that with this example here, in which A is the Angustifolia alleles of the nuclear gene, and L represents the Latifolia alleles of the nuclear gene. So if we take this cross here, F1 times F1, producing an F2, of course, and do a simple, and these are the alleles that are present in the F1s. They have one of each parent species. And just do a simple Punnett square, you can see these are the possible outcomes in the F2s. So, and we know that they will have Angustifolia chloroplast DNA because, as I mentioned before, Angustifolias are always the maternal parents of the hybrids, or almost always. So will we be able to identify these from PCR RFLP? Well, these two, yes, based on the nuclear gene, because they have one of each species. This one, yes, because it will identify the nuclear as being Latifolia. And if we do it in concert with the chloroplast DNA, that will be identified as Angustifolia. So we'll have cytonuclear disequilibrium there, and we'll be able to identify it that way. The fourth option we won't be able to identify because it will appear as Angustifolia based on nuclear and Angustifolia based on chloroplast. So we would be able to identify about 3/4 of these hybrids. But we also have to think about how likely are the different crosses. So we can do the Punnett squares and figure out, what we would expect to find. And then we have to look, okay, what's going out in the wetlands on the landscapes? How likely are these different crosses going to be? And that's based on both the viability of different crosses and also on how common different plants are. And actually, Angustifolia is much less common around here than either Latifolia or hybrids. So what that means is any cross involving Angustifolia should be less common. And of course, as I've talked about, some of the back crosses and interspecies crosses are asymmetrical, so that also limits things. This one should be probably the most common type of hybrid because there's lots of Latifolia around here and there's lots of hybrid around here. And for these ones, if we do the same approach with the Punnett square, we're crossing here Latifolia times hybrid, an F1 hybrid. And these are the 4 different genotypes. And combined with the chloroplast, which is the Angustifolia again, because the hybrid is the maternal parent, we would in fact be able to identify all of these. These two just from the nuclear PCR RFLP, and as before, these two by combining the nuclear and the chloroplast RFLP. I don't have time to explain all the rationale, but this table just shows, based on the different cross types in this hybrid system, if we use PCR RFLP from both nuclear and chloroplast, again, really quick, easy test to do, this is the proportion of hybrids that we expect we would be able to detect. And this is how the expected relative frequency of the different cross types. And you can see that the common ones, we should be able to detect them 75 to 100% of the time. The main takeaway here is it's really important to understand the biology of the system that you're trying to come up with these quick tests in. And that's based on both the reproductive biology, but also what's happening on the landscape in terms of relative abundance of different taxa. Now, this test is I think it's going to be very useful. There is a caveat. It's not the kind of test where you could take one individual plant, run this quick test and say, yep, that's a hybrid, that's a backcross to latifolia, or that's an F2 hybrid. It's not going to give that level of detail. But for site level characterization, it's going to be really useful. We're talking about a very big geographical area. And sometimes we need to know from different sites if the invasive hybrid is in there. For example, there's a picture of a muskrat here, because I'm aware of an ongoing study that is looking at a whole bunch of wetlands around the Great Lakes region. Muskrats do very well in wetlands that are dominated by Typholatifolia. There's some thought that they maybe don't do so well when the wetland is dominated by the invasive hybrid. And so to understand that, people need to survey lots of wetlands. They can count the muskrat houses. How do they quickly assess whether there's a high proportion of hybrids? So a test like this will be very useful. It will also be useful for looking at things like range expansion. So this is a paper that's currently under review, in which I collaborated with some researchers, mostly in the States. And we collectively, we basically pooled our data from different parts of the prairie pothole region. From this region, previously, there were very few reports of either angustifolia or hybrid. And what we found, to our surprise, is there's actually more hybrids in this region than anything else. And this region is very important for migratory waterfowl. The so-called prairie potholes are important wetland sites, both for waterfowl passing through and also for breeding birds. And so there is a bit of a concern as the hybrids spread over this landscape, what knock-on effects that's going to have, how the landscape might change. And so again, these quick tests to go over these very large areas, such as the prairie pothole region, can be exceedingly useful. So sometimes old school can be better. You don't necessarily need to use an expensive and fancy molecular test to get some really useful information. Now I'm going to talk about something a little less old school, and this is early detection of invasive aquatic plants. So again, coming back to this photo, this is a big mat of floating yellow heart. Another point to make around these types of images is once there's this degree of establishment of a new population, it's very difficult to manage. So key to managing invasive species is early detection. The sooner you can realize that they're there, the much easier it will be to prevent them from, really getting hold of the habitat. And in theory, one way that we can do this early detection is through environmental DNA. And I'm sure everybody in the audience is familiar with environmental DNA or eDNA, which rests on the principle that organisms always leave traces of themselves in the habitat in which they live or in habitats through which they pass. And these traces will very often include DNA. And so that's what we call environmental DNA. DNA that's in an environmental sample, it's no longer within an organism, but it's been shed in some way. And it's very easy to get environmental DNA. In this example here, this is how you would get environmental DNA from a water sample. You just collect the water, filter it, all of the particulate matter collects onto the filter, and then you just use a kit to extract the DNA from the filter, so you're separating DNA from everything else. And then you end up with a sample of environmental DNA that collectively represents, in most cases, many different taxonomic groups. Once you have done that extraction, there's two general approaches you can take. Because it includes DNA from lots of different taxonomic groups, you can either use a barcoding approach to try to characterize DNA from a particular species, to understand, for example, if the species is there through presence or absence of the DNA, or you can use a metabarcoding approach, which simultaneously characterizes DNA from multiple species. So those are the two sort of broad strokes, general approaches you can take. These two figures are from the reference that's written at the bottom of the slide, and I just want to make 2 points from here. One is, as you can see on the left, the numbers of published eDNA studies has been skyrocketing in recent years. It's a new field, but it really is taking off. More and more studies are being published on this, ever increasing interest. The figure on the right shows that most of these studies have been done in freshwater systems. And a big part of that reason is it's a lot easier to get eDNA out of water than probably anything else. If you look below that, you can see that a lot of these studies have been done on fish, invertebrates, a lot of them there. And this is my addition to this figure, just to illustrate the fact that aquatic plants have actually been really understudied. When you consider how much, what a high proportion of eDNA studies have been done on freshwater systems, you might expect aquatic plants to make up a reasonable proportion of these, but actually, this is my estimate, but there's been very few, surprisingly few studies done on aquatic plants. So this begs the question, why have there been so few aquatic plant eDNA studies? Is it because people aren't interested, they don't think it's useful? And no, I don't think that's the case. As I've already mentioned, this can be a really useful approach to rapidly screen multiple sites to look for early detection of invasive species, for example. It can also be used to describe plant communities. It can be used to screen multiple sites to look for species at risk that might be difficult to locate using more traditional types of surveys. So the problem is not disinterest. I suspect that the issue lies more around technical challenges, and associated with technical challenges is publication bias. And by publication bias here, I mean, it's a lot harder to publish something that does not give you a nice, clean, easy to explain result. Or if you're reporting results that can be applied with some confidence. When there are question marks, when there's inconsistency, it is a lot harder. And I think that publication bias is often linked with technical challenges. And I'm going to come back to this at the end, but I think this is feeding into some of the reasons why there are relatively few aquatic plant eDNA studies to date. For a few years, we've been looking at eDNA as a tool to use in association with an invasion by the water soldier, Stratioides alloides. So again, this is a plant that is of interest in Europe. It's native to Europe and parts of Europe and parts of Asia. It's a species at risk in some areas. Well, there is one invasive population in North America that has a lot of people worried, and it's about an hour from here, about an hour from Trent University, which is where I'm at. And it's a problem. It is dominating a habitat, and it can spread very easily. And this is why early detection is important. The invasive population near here, as far as we know, it does not produce sexually. Very few flowers have been documented, but the reproduction through clonal offshoots is prolific, and it also produces these turions, which are vegetatively produced propagules that can overwinter. So early detection is important, partly because of this strong facility to asexually reproduce. Also, the biology of many populations is interesting because in the autumn, the plants will often sink to the bottom, and they can overwinter on the substrate as rosettes or as turions, which can embed into the substrate. And then in the spring, they rise and start growing. So if you're doing surveys at certain times of the year, the plants may be there, but they might be very difficult to see, particularly if the water is quite murky. So for all of these reasons, and the other reason why early detection is important is, so this is the invasive population I was telling you about. It's in the Trent River, and it's actually in a section of the river that widens out. It's sometimes called Seymour Lake. It's not a lake, but it's a wider, shallow, more stagnant part of the river. And this map here is just showing you, so the map on the left gives you the location in North America or in Canada of this site. The map on the right is just making the point that this is part of the Trent 7 waterway. So the river in which this population grows is on the Trent 7 waterway, which is a few hundred kilometers long. It connects Lake Huron to Lake Ontario. And of course, we do not want this invasive plant to get into Lake Ontario, because then it could be all of the Great Lakes and into all of the different watersheds. So that is a concern. But you can see from up here, it really does change the landscape. It is problematic. So this was our question. Does eDNA provide a useful tool for water soldier surveillance, both for identifying newly established populations, and also because there's a lot of attempts to eradicate this, could eDNA be a useful tool to test whether or not eradications have been successful? Well, and this is definitely a shortened version of the story, because there's been multiple projects spanning multiple years, but the important parts are here. We began by collecting samples from the Trent River, both in the summer and the autumn, so in July and in September. And we extracted eDNA from these samples, and we tested with markers that we had designed to be specific to water soldier, that targeted a region of RBCL from the chloroplast DNA. And we had two sets of primers, one amplified, a fragment of 437 base pairs, and then one that we later used targeted a smaller fragment of 107 base pairs. So we collected a bunch of water samples, extracted the eDNA, and then did the assays using these markers, qPCR. And this is what we found. The samples that we collected in July, even though, just to remind you, this is from a site where there's a lot of these plants, we didn't get any water soldier eDNA detection. In September, roughly half of the samples revealed water soldier DNA when we were targeting the 437 base pair fragment. And then when we later redid them using 107 base pair fragments, a much shorter fragment, we found water soldier eDNA in close to 2/3 of the samples. The average number of copies was comparable between the two. So the conclusions based on these qPCR investigations are that, yes, there's water soldier eDNA in these water bodies. It does shed eDNA, but detection is inconsistent, even though there's a lot of plants there. And these data suggest that the timing is important. So maybe not collecting samples during peak growing season, but waiting till the plants are starting to senesce in the autumn. Which is, in hindsight makes sense, because they should be shedding more DNA at that point. And also the type and number of qPCR assays is important. So we did have more success with a shorter fragment length. And this is also because even with the short fragment length, even when they were sampled in autumn, we still got only about 62% of samples gave positive. So this is highlighting the need for, quite a lot of replicates before you can be sure. So is this useful for surveillance? I think some utility, but it's certainly, by taking this approach, you're not going to be able to say, yep, there's water soldier in there, or no, there's not water soldier in there. So it can be useful, but it needs to be done with understanding around the limitations of this process, and would probably be most useful designed as part of a more holistic approach to monitoring these sites. And then I'm finally going to talk about, this is the only part of this seminar that's been published. It was published earlier this year, so I won't give you a lot of detail. You can see this, read this paper. It's open access if you want more information about it. But after the qPCR gave not the results we were hoping for, we did develop a metabarcoding assay that was targeted at communities. We designed primers targeting two chloroplast regions, MAC-K and RBCL, and also the nuclear IDS1 region. We tested on samples from two locations. The first was the same one that I've just been describing, the Trent River location. I just highlighted this. You can see that these were sampled again in the summer, in July 2017. I'll come back to that in a minute. And then we also collected from another location, Blank River, which is a place where water soldier has been identified, but in that case, they got on it really quickly and it has been hopefully eradicated. as of around 2017. We found that of the primers we designed and the regions that we amplified, the most effective were a pair of RBCL primers that amplified a fragment of around 220 base pairs. And this identified 20 species or genera, usually to species, a couple we only identified to genera, across 5 sites at these 2 locations, Trent River and Black River. And of the species that we identified, species and genera, 9 of them were alien taxa, which I'll just show you here. These are the 9 alien taxa. And you can see here are the names of the species, or in the last one, we just identified it to genus. And the red lines take you to Trent River, and then the blue lines over here take you to Black River. And note that of the 9 we identified, one of them was water soldier, even though at Trent River we had collected the samples in July 2017. So this suggests that this is actually a much more sensitive tool for assaying plant eDNA compared to qPCR, because even with far fewer samples, and even when they were collected in July, we still picked up the signal of this invasive species. So that was really interesting. What do these positive detections mean? Well, as I mentioned, we identified 9 alien taxa in total. 6 of them were not surprising. They've been previously detected either in the water bodies from which we sampled or in nearby water bodies. 3 of them have either not been detected nearby or have been detected only in the summer. It's thought that they have so far been unable to overwinter, although that's likely just a matter of time. But all of them are established in the northern USA. So there is range of established populations of these extends to just south of the Ontario border, and they're all on Ontario's invasive species watch list. Now, one of the challenges with the eDNA, of course, if you get a signal, it doesn't necessarily mean it's an established population. It could be an ephemeral signal. When it comes to invasive plants, people dump their aquarium tanks into the lake, or they clean out their back garden pond. So there can be transient signals in there. It doesn't always mean that there are established populations. And this isn't this isn't a limitation specific to metabarcoding. It's specific to all eDNA studies. But to me, this is certainly a flag that at these sites there needs to be more surveillance if we are going to catch these early colonizing invasive species. So in conclusion, I think it's safe to say that eDNA work is harder to do on plants, or at least the small number of species of plants on which it's been tested. In many cases, it's harder on plants compared to other taxa, like in fish, for example, it seems to be a lot easier. It can be effective for detecting plant species of interest, but experimental designs need to factor in things like the timing of collection for at least some species, whether they're growing versus if they started to decay at the end of the growing season. As I mentioned, needs to factor in the species of plant, thinking about the morphology of the plant, the biology of the plant. Some of them may decay much earlier in the season. As I mentioned, few have been investigated. Also needs to factor in the type of assay. When it's qPCR, consider the fragment length, consider the number of replicate assays you need, and metabarcoding, although it does seem to be more sensitive, at least with water soldier, it does require more specialist skills, although the good news is it's getting much cheaper and more people are acquiring these specialist skills, including the bioinformatics. So that is becoming more accessible. But future work, I think that publications on eDNA assays need to include more of the ones that didn't work. And I put work in quotes because you can have results that maybe aren't what you expected, but it doesn't mean necessarily that this is not a useful tool. It just needs to be, we need to realize what the potential limitations of the tool are, but it can still be useful. Even if you don't get any result, it can often be useful to publish, because otherwise you can get lots of people in different labs that are doing the same thing, not knowing that other people have already tried this. So I do think that we need to be a little bit more transparent about difficulties that we have with characterizing plant eDNA. And then that leads into the second point here. Once we know what is working, what is not working, or what is leading to unexpected results, we can then design more informed experiments that will directly compare different approaches instead of doing what I did today, which is kind of taking a post hoc retroactive kind of view. And then the third recommendation for future work, of course, is coming back to this idea of what does an unexpected detection of alien species mean, thinking about how you would then follow up. You know, we have a result, oops, bit of a flag here, it could be in there, thinking about most effective ways to go in and maybe do surveys, timing of the surveys, thinking about habitat, subdivisions within sites, and so forth. Okay, I may have gone over time. I did cover quite a lot there, and I'm just going to end by again reiterating that a lot of other people contributed to what we did here.
All right, thanks very much, Joanna, for your very clear and interesting talk. Unfortunately, we'll have to move on quite quickly because the next speaker has a meeting, an event afterwards. So we'll make room for questions after Anthony's talk, if that's okay.
Sure, yeah, I'm sorry I went over. I was probably covering too much.
No worries, no worries. So I'll share my screen again. Did I share my screen? Okay. So our last speaker of today is Anthony Booker. He's an expert in Chinese herbal medicine, so medicinal plant science at the University of Westminster. He's senior lecturer at the university. He published in a wide variety of scientific journals and books, and his current research interests focus on the use of plant metabolomics and how this can be used to improve the quality of traditional medicines, thereby tackling issues such as adulteration. In his presentation today, Anthony will discuss the up-to-date analytical methods for herbal medicine authentication to combat illegal practices, such as adulteration. He will further focus on the need for standardization in the regulation of the global supply chain. Anthony, the floor is yours.
Okay, thanks very much. I have to go through this fairly quickly, so I'll skip any introductions and go straight in. So, analytical methods for herbal medicine authentication and the need for standardization in the regulation of the global supply chain. So, we've had the introduction of a European use registration system that's been looking at the quality of herbal medicines, and that's really improved the situation in both the UK and in Europe and with regarding quality. However, this only really affects traditional herbal medicines and ones that are registered. So there's still a major problem with food supplements. And often the reasons underlying the poor quality and the adulteration within food supplements usually comes down to people wanting to make more money and more profit by using cheaper ingredients or adulterated ingredients. So most of our work has been looking at detecting these adulterations and poor quality across the sector. So in this presentation, I'm going to look at some examples using different analytical techniques, how herbal products, including food supplements or nutraceuticals, are adulterated, and mainly by looking at their phytochemical fingerprint, either using mainly HPLC, but also with NMR. Just before that, I'm just going to explain that we can separate herbal manufactured products into three broad categories. First of all, products that are generally considered to be foods. Secondly, products that are generally considered to be herbal medicines, but may also be legally placed on the market as food supplements or nutraceuticals. And thirdly, a product that can only be legally placed on the market as a medicine, either with a full product license or as a traditional herbal registered product. In America, most products are regulated as foods. In Germany, most plant products are regulated as medicines. In the UK, we stand somewhere in between, where some are food, some are medicines, and some are traditional herbal medicines. So firstly, we'll take a look at products that are considered, or take a look at a product that's considered to be a food. So we can look at turmeric, curcuma longa. So used as a food, but also has a long history of use as a traditional medicine, both in Asia and in Europe. And there's quite a lot of medicinal research that supports its use as a traditional medicine, or as a anti-inflammatory, or to aid for digestion. And research has also indicated that the active ingredient, the curcumins, could also be used in the treatment of cancer. The main problem with the curcumins is that they're very poorly bioavailable. So there's a lot of research into looking at how to improve bioavailability and how to use different dosage forms to improve absorption, such as using nanotechnology. So we looked at different samples taken from that I either took from India or from the internet or from health food shops, and we run HPTLC to look at the difference. One of the main differences we were looking for is for adulteration with a related species called jungle turmeric, commonly known, which is Curcuma aromatica. And if we see the bottom three bands are the three different curcuminoids, which is curcumin, dimethoxycurcumin, bis-dimethoxycurcumin. If you see to the left-hand side, the first 1, 2, 3, 4, 5 has that very bottom band missing. So it's only got 2 of the 3 curcuminoids. So this is a very nice way of differentiating between curcuma longa and curcuma aromatica. And they often not curcuma aromatica is used to, it becomes an adulterant, and it's used to improve the colour of curcuma longa. Further to the right you see with the very dark red bands, which are very high levels of curcumin. These are strong extracts. But if we go from 1 to the third one in, it seems only to be contain curcumin and nothing else. And this is highly likely it's just been the product has just been spiked with pure curcumin. And the ones later on contain very poor curcumin, and the ones right at the end, the last 3, are actually Chinese herbal extracts. And the problem with Chinese herbal extracts in this context is that they're normally aqueous extracts, and because curcumin isn't soluble in water, you don't get any curcumin, so they're not really very useful as products. The products that we looked at, we found we compared with the fresh rhizome. So we took fresh rhizome and dried it and analysed it, and that's in band 39 and 40, and you can see some extra bands which I've circled, and these are tumerone, which is contained within the essential oil, and it's what gives turmeric its very strong smell. Now the problem with tumerone is because it's volatile, it's soon lost. So you can see with different products, they don't contain this tumerone band, apart from the ones in 47, 12 and 13. Now, the difference with those products is that they were part of an vertically integrated value chain. So the contract was already made with the farmer. The turmeric wasn't going to auction, so it was going directly to the farm, to the manufacturer. And because of this shortened the supply chain, and so the volatiles were preserved in a much better way. Yeah. Okay. Moving on. Okay, secondly, we can look at products that are generally considered to be herbal medicines, but they could also be legally placed on the market as food supplements. And this is mainly dependent on what you claim. So if you put, for instance, garlic on the UK market and make no claim, it's a food supplement. If you put garlic on a shelf and you say it helps sore throats, then you need either a full medical license or a traditional herbal registration. So purely on the basis of what you claim your product can do determines its regulatory status. So the one we're going to look here is Rhodiola rosea, often known as golden root, herbal medicine that's used to its traditional herbal registration is it has an indication to treat stress-induced fatigue and anxiety. So that's a allowable claim that it's allowed to be made as a traditional herbal medicine. It's got traditional history of medicinal use within Eastern Europe, Scandinavia, the Arctic countries, Asia, North America. All above the equator and mainly in high altitude regions. So again, we looked at different products, taking many from the internet. We mainly trying to differentiate the ones that contain rosavin and ones that contain salidroside. Most rhodiola species contain the salidroside, but it's only Rhodiola rosea that contains the rosavin. And it's the rosavins that most of the research has been carried out on. So and this is why Rhodiola rosea is more expensive and more highly valued than any of the other rhodiola species. So again, out of our the samples we looked at, you can see at the bottom, number 11 was a reference sample. So that's what Rhodiola rosea should look like. Rosavin in the first two columns got salidroside and rosavin, so you can see that's really the only one that matches up with 11, apart from 20, but it's a lot weaker. So all of these were regarded as poor quality samples, and number 8 appeared to contain nothing at all. Number 8, in fact, although it claimed to be Rhodiola rosea 2,000 milligrams wild source from Siberia, it didn't contain any rosea or any Rhodiola rosea or any rhodiola species. And when we did further tests using mass spectrometry and NMR, we found it was actually it contained 5-hydroxytryptophan, which is a mild natural antidepressant. So people probably may have felt quite good taking it, but they wasn't taking what they thought they were taking. We did we looked at some other products and did a program for a BBC program called Trust Me, Doctor, where we looked at exactly the same issues and do herbal supplements contain what they say on the label. One of the products we looked at was ginkgo biloba, which has a traditional herbal registration for Raynaud's, although most products are on the UK market as food supplements. So this is a case where it's both a THR and a food supplement. We looked at the different ginkgo products. This time we looked at various products, and we found our standards were rutin, chlorogenic acid and quercetin. And if you look at the SST, the bottom one is rutin, and we found apart from some being very poor quality, this number 7 was the same company. It was 5-hydroxytryptophan again, so being sold as ginkgo this time. But otherwise, we had varying quality samples, but what we did notice is some were very high in rutin. And then we surmised the reason for this is that the test, the pharmaceutical pharmacopoeia test is, or the test claimed is total flavonoids, often of 24%, and rutin is a flavonoid. So if you've got some ginkgo and you want to use less of it, you can just add rutin. This is what the companies were doing. And if you do a test for total flavonoids, it will hit your 24%, but it won't be due to the ginkgo flavonoids, it will be often due to the added rutin. So this was a case of, you know, manufacturers trying to boost their product through adulterating it with cheap ingredients. So rutin and quercetin, one being the rutin is the glycoside of quercetin. So out of the 35 samples, only 2 samples complied with the European Pharmacopoeia, and many were contained elevated levels of rutin or low ginkgo metabolite content. Over 50% of samples were found to be adulterated, containing elevated levels of rutin or incorrect species, for example, Sophora japonica, which has a very similar chemical profile, or has low ginkgo metabolites. So either being adulterated or being produced using poor extraction techniques. For the BBC programme, we also looked at milk thistle, looking at the different flavonolignans. And once again, the ones on the left-hand side were the first 2 were standards, the next were registered THR products, and the next 1, 2, 3, 4 were just food supplements. The first one seemed to contain nothing at all. The next 2 were very low in poor quality, probably because they didn't use high enough alcohol to extract. And the last one didn't appear to be milk thistle at all. So again, 36% were adulterated or of poor quality. We also looked in this case with NMR. NMR, you can see a much broader range of components and you see varying quality, but the top 5 we could see contained almost nothing at all of the milk thistle. And then finally, a product that can only be legally placed on the market as a medicine or a THR, so it doesn't matter what you claim because of its pharmacological action. It's regarded as a medicine, and this is St. John's wort, Hypericum perforatum. Again, we looked at a range of 47 samples. In this case, we found that the American samples, I should say the North American samples, came from different parts of the USA, and we found they contained artificial dyes, such as sunset yellow, tartrazine, brilliant blue, etc. And at first we couldn't work out why this was. But with some closer investigation, we found that they were added to elevate the total content of hypericin, and this is analysed using the United States Pharmacopoeia method, and the dyes absorb at the same wavelength, and so the total reading you get is higher. So again, you can use lower amounts of St. John's wort, add some artificial dye, and it will pass the United States Pharmacopoeia method. Perhaps I shouldn't be telling you all this, but this is some of the tricks of the trade, if you like, people resort to save money. So in conclusion, herbal medicines, especially food supplements, vary greatly in their phytochemical composition. Some herbal medicines and their starting materials are known to have been adulterated with either incorrect species or have chemicals added in an attempt to deceive analysts. HPLC is an efficient and economical technique used in the quality control of herbal medicines. Thank you for your attention. I think we got through that okay.
Thank you very much, Anthony, for the talk. Interesting. It's kind of worrisome that so many products are like being adulterated and are still ending up on the market, on the herbal market.
Mm-hmm.
So yeah, if anyone has questions, now it's the time. If Anthony, you have to leave soon, right?
Yeah, I'm okay for a few minutes if there's any questions.
Okay. We have a Feli. Feli, you have a question?
Yeah.
Yes.
Question in return, I guess. So, Anthony, in how far does your scientific outreach really bear fruits in like the private market and sector and the supply chain to Europe?
Bear fruits.
Yeah. Did you say that? Is that an expression? I don't know.
So what do you mean? What impact does it have? Okay. Well, with all these investigations, we make a conscious decision not to name specific companies' names. You know, when we publish papers, etc. But we do pass the information on to people like the MHRA, which are the UK medicines regulators, and the Food Standards Authority, who look after food supplements. And then we leave it up to them to take any action. Now that they are very kind of understaffed, and whether any action takes place, they're not very proactive, and they only react when there's some kind of complaint. But I think through our analysis and through the results we've presented, you know, some of these companies have been investigated. So that's really the kind of impact we hope we're going to have.
Nice. So you address also manufacturers directly and respond.
We pass the information on to the regulatory bodies and let them handle it. Yeah.
Okay.
Okay. Are there any other questions for the audience? No, apparently not. Okay. Please, we do thank you very much, Anthony, for presentation, for being here. And so if you just all want to, because this is the end of the symposium series, if you would leave your camera on or turn your camera on, so we'll make a group picture of everyone. Just 1 minute, we have some technical issues. Perfect. Okay, so 1, 2, 3, smile. Here we go. Okay. Thank you very much for being here and for to have made this possible. It's also the last Plant.ID event, by the way. So yeah, on behalf of all the PhD students, I would like to thank also the whole Plant.ID consortium. Yeah, to have made this possible. So thank you very much. It was a pleasure to be your host, and thank you to the speakers as well. Good.
Yeah.
Have a nice afternoon. Bye.
Cheers.